So Sarah, thanks for coming today to talk to me.
It's great to be here, thank you.
So we're going to talk about the work that I made as part of we began as part of the Body Project, which started obviously when I was doing the Artist in Residence at your lab in 2017, which was organised by ASCUS Art and Science. So can we start off by you explaining a little bit more, a bit about your research?
Yeah, so my research focuses on this skin disease, eczema. That's an itchy skin disease. It affects many, many people, and it ranges in severity, as you know, from something really quite mild to a disease that can change somebody's life. And the approach I take is trying to understand the genetic mechanisms that control how your skin's made and make you more or less likely to get skin inflammation. And as you know, we use skin cells in the lab. So these are cells from human tissue that we culture to make artificial skin, so that we can really, change what's happening inside the cells to really get to the root cause of how the DNA controls how the skin is made and how it functions. So it's very human research, it's human tissue that we're using, and it's a very human condition. So I'd always been keen to explain that to people, to help people understand what skin is about and why my research is important. But I thought, often people are not allowed to come into the lab. So I was looking for ways that somehow we might be able to explain or reveal the lab in new ways to people outside. And inviting an artist in seemed a great opportunity. So how did you find it? How did you get started? Or tell me about how it felt when you started in the lab and what approach you took.
Well, I found it fascinating, and it sort of adds to because you were already working with public in some ways through science public engagement, weren't you, anyway, as well? And I am interested in that as a way that I work that sort of adds to science public engagement quite sort of specifically, but in different ways from you might have done it before. And it was a fascinating environment. I mean, for me, that first, because it was about 6 months that I was there 1 or 2 days a week, wasn't it? And so what I found was really useful was the way that then you allowed me to observe what was happening, and it was a huge generosity of just letting me be in the space. So I worked quite closely with Sheila, your technician, as well, and sort of working really observing the processes, so actually practically, but there was also lots of more sort of philosophical and ethical conversations. And then it was also coming to the seminars that you did, so the ones that you would do that were either your own, or then also sometimes having people from other labs, and then inviting me to talk as well, which was really lovely as a sort of exchange that was happening. So it was, and then what I think was building through this, so that's quite typical of how I'd work, where I'd go into a situation and observe first of all, and then the thing that I really sort of started to attach to was the artificial skin itself, the organotypic, or you call it organoid as well.
You can call it either, yeah.
Yeah, so that in itself, I was really fascinated about the way that the level of care and attention that was extraordinary that goes into it, and the precariousness of this 3-week-long life cycle from theatre to the lab to then disposal. But then also the way that you would all talk about it as well, in terms of I was really fascinated. And there was also just the point that actually it's human tissue, but it's outside of the body, in vitro. Is that the right term?
Yeah. In vitro.
In vitro, and and the whole idea that then it's human and living, but no longer within the body. So it sits in this sort of paradoxical situation.
And is that kind of what you expected? I mean, how much of that did you know before you went in the lab, and how much was a surprise?
Well, I think I was a surprise, but it's what I'm interested in anyway, because I'm interested in the relationship between the body and science and technology, and what the sort of expansion of these things adds and shifts in our perception of what it means to be human, what it means to our lived experience. But what I was surprised about here was the lived experience that I focused in on was actually the cells, not so much. I mean, I was really interested in the staff as well, but I focused on that as this other version of human that was happening in the lab. And what I was really curious about as well, so it's the process of analysis that you were using, and all these very, very detailed processes. But then also the way that you all spoke about it, because you used phrases like whether the cells were happy, didn't you? I mean, that's quite a common way, isn't it?
Yeah. We talk about the cells. I mean, they are a living thing, so you do need to keep them happy, just like you would a baby or any living creature. You have to give them what they need and make sure you take away what they don't, what can harm them.
and it was that fragility of that process, but the care and attention that was going on from temperature, from feeding, from everything, all those layers, that was fascinating. And that's where, and it was actually through the talking that I was actually, doing talking to you all a lot. And that's where I decided to actually then interview. It was 3 of you that I interviewed on the process and the journey of the cells from the theatre to the lab. And theatre, when we mean like a medical theatre, as an operating theatre, theatre to the lab, and then to disposal. And then what I did was crafted that into a script, which was then, but then what I did was I took that information and wrote it from the point of view of the skin cells, because I was interested in their experience. So it was shifting. So, and what I did, if you remember, I checked with you quite thoroughly about the script as well, because I wanted that it was 100% scientifically accurate, that the whole thing is accurate, but also had this more emotive feel to it through the descriptive phrases that you used.
And that's where your skill comes in, because obviously I can write things that are scientifically accurate, but taking that different perspective or expressing it from the perspective of the cell was not something that we'd ever thought about, really.
Well, and it's not what would typically be published in your way, isn't it, in your world? So that's what I was very interested in that and very much the sort of work that I'm interested as well, in terms of that emotive, how you can bring emotive to science and technology and bring different ways for people to understand what's happening, and think about the ethical and practical and philosophical issues around those situations. So then, if you remember, I used the speech synthesis voice, Heather, which is the only Scottish speech synthesis voice, cool, to then sort of bring the script to life, which is then become a key element of the exhibition here, because that's a key part of the soundtrack for both the film and the AR, the augmented reality app. And then I think following that, it was also then working quite closely with yourself and other staff about actually to create these, the 3D printed skin cells. So these were you'd very kindly allowed me to use a sort of extra of your cultures, wasn't it? And then do you want to talk a little bit about that process? Because what I used was confocal microscopy, wasn't it?
Yeah.
And do you want to talk a little bit about that process, how it fits in with the lab?
Sure. So we grow skin cells all the time in the lab. Like I said, we use keratinocytes, the cells are called, extracted from real human tissue. And obviously we have consent from patients that kindly donate their skin when they're having surgical procedures where skin might be removed and it would otherwise be thrown in the bucket. They get, with their approval and their consent, they allow that skin instead to be brought to the research lab. So then we can extract the cells. We culture them in a pink liquid that allows the cells to grow. And keratinocytes, the skin cells, always want to kind of join together, settle down and form layers, because this is how the skin's made. The cells very much join together and then grow up into layers. So the tissues that we gave you were exactly that, skin cells that had been allowed to settle down onto a surface and then start to form layers. And it was Martina that helped. She labelled the cells with antibodies that are specific proteins that attach to another specific protein. So we all know, antibodies react against infection, but in the lab we use antibodies to react against a special part of a cell. So you can tell the antibody you can design an antibody that binds, that sticks to either the centre of the cell, the nucleus, that's what these are, or the skeleton of the cell, kind of literally the structure of the cell to show the shape, and that's what these are. And the antibodies are proteins that have a fluorescent tag on them. So then when we shine light on the fluorescent tag, you can see where the protein is bound. And most times most microscopy that you see is two-dimensional, but we can make three-dimensional images using what's called confocal microscopy, because that's a technique where it focuses at a specific layer in the tissue. So not just cutting through one section, but focusing at different layers and then using those images to reconstruct a three-dimensional shape. And that's what you were able to do with the skin cells specially labelled to show different parts of the cells, and then the confocal microscopy to layer it up to make a 3D image. And I know it was Paul that helped you with that, wasn't it?
Yeah, it was Paul Appleton, because for me as well, what was really interesting was it was like a bridge, working with the confocal microscopy. It was a bridge from early research that I'd done when I'd first come in, where I'd gone into like the collections as well at the University of Dundee, and there was some stereoscopic imagery in there as well, which was that classic of that actually that you could only really use within a stereoscope now, but had been used previously as teaching aids that would be projected as three-dimensional imagery. And that I was really interested in that, alongside there was 2D illustrations, which would be more the classical things, and then things such as this 3D model, which is then from your office, isn't it, which you very kindly loaned us to put into the exhibition. But then for me, the confocal seemed to sort of add this literally like another dimension and possibility to how to look in more precise ways at that, whereas a lot of the stuff that I'd been looking in the collection seemed to be seen more homogenous, whereas actually with the confocal you could really see the sort of individual nature of the cells very clearly. And it was working with Paul Appleton in the imaging facility to then try and find a way, because it wasn't a typically used way, although they had these slices and they would build into a 3D shape potentially digitally. It wasn't something that was typically done where that was actually taken out into sort of a CAD type file, which actually, as Paul and I worked on it, we did then find there was something was released that basically did it right at the end of the process after we'd gone through it lots and lots, doing a pipeline of. But it was a really interesting process of trying to take that three-dimensional data from the confocal microscope and actually then take that into a CAD style software that would then 3D print it, and that's the results here.
So Bev, it's a classic example of an artist asking difficult questions, which you did repeatedly. I don't know if I've ever told you this, but I asked Martina to remind me about the green labelling, and we were both reminiscing about the questions that you came to us with. How can we do this? Kind of look at the cells in this way, and we were constantly tearing our hair out, trying to understand the artist and give you what you needed. Because the way we use confocal, it does make 3D images, but we tend to use it to understand the 3D structure by looking at it in different layers rather than putting them all back together. So you're absolutely right, the CAD file, that's what we had to extract.
Yeah, so that was the different way. And then the sizing as well was that these are actually 2,000 times larger than life. Because obviously in microscopy, you're looking at them on a larger scale, isn't it?
But
and I think it was,
you don't get a feel for it.
No, and it was to try and give them that handheld physical relativity so that people can see that they're obviously clearly magnified, and it's part of the exhibit that it will say it's 2,000 times larger than life, but that also people get an understanding. And I think and we had some really interesting conversations about this as well, about how then I'm displaying them, which is because, I was really struck as well from my sort of way more limited knowledge of scientific illustration, but how homogeneous things normally would seem, and then how individual, and really understanding that the cells are individual in themselves, but then there's also the going through differentiation as well, which is what I've tried to, in the larger exhibit when that's on, it'll show in the process that that's done as a horizontal way. But can you explain?
So you can see, so these, as I say, the green stains the whole shape of the cell, because it's like the skeleton that gives the cell its 3D structure. And you can see just by looking at them, they're very different shapes. So the reason for this is keratinocytes, or skin cells at the base of at the deepest level of skin, start off being quite plump and round. And then as they divide and move up through your skin to the outermost layer, you can see they get flatter until the very surface cells are really quite flat. So these would be quite close to the outermost layer of the skin. And then even after this, when we develop the full skin model, even after this layer, the cells get even flatter again, and then they can shed off the skin surface in real life. So that's why they don't look the same. It's because they're going through different stages of development or different stages of differentiation. The nuclei, so this would be what controls the cell, has the DNA inside it. The nucleus of the cell would be deep inside. And again, even the nuclei are different shapes. Some of this is an artifact, the way you're, really just seeing it in one second, split second of time. But the nuclei do vary in size and shape, and as the cells reach the skin surface, the nuclei are destroyed and lost. For me, I spend my life studying skin cells, but to see them as a 3D object, you do see them in a different way, and it's quite nice that it's 2,000 times life size. It's pretty mind-blowing.
And you have a set that you use now, don't you, in science public engagement as well, which was a really lovely sort of loop that made as well, that it's actually been used in both. It's both been used in exhibitions, but it's also been used within science public engagement as an extension of the lab's work.
It's a great way to talk to people about skin cells, to say, here they are. Yeah.
And then as a follow-on from that, then I was interested really, because I found the whole sort of situation so fascinating that I really thought about that early sort of parts of it as initial research objects. And then I was interested to continue it, as then we sort of kept that conversation, although I wasn't working in the lab so much specifically at that stage. We were still in touch, weren't we, about actually continuing the work. And what I was interested was really to add, which is very much part of the featuring in the exhibition at Summerhall, is to add ways to bring an audience into the lab, but in ways that were not necessarily about a sort of didactic, instructional, informational way, but that sort of trying to enable people to think about those more sort of poetic, ethical questions that the lab raises, because it's very complex what's happening in the lab, in both practically and in terms of those sort of ethical scientific questions that are happening. And so I was really the extension there, and it was very much thinking about the fact that people can't really, as you've said already at the beginning, it's very difficult to bring people in, isn't it? I mean, just to say the practical considerations.
Yeah, I mean, there's all health and safety. Obviously, there's a lot of dangers within a lab space that you can't just allow people to walk through. The equipment that we use, the potential risk for infection. So dangers to anybody passing through the lab, but also obviously dangers for the equipment that's in the lab. We have a lot of very expensive, very sensitive equipment. So yeah, we rarely invite people into the lab. Even when we do open doors days, you know, it's still only a very limited number of people that are allowed to just walk through the lab space supervised. So yeah, you were really in a privileged position to be allowed pretty much free rein to come and go and chat to people. Obviously, recognising health and safety, and you were very good at not fiddling with stuff. Bev didn't break anything.
Because it is even an infection as of the actual cultures themselves, isn't it? But not only for us to potentially get something, but us to potentially infect the cultures. And that, I think, was really sort of how, again, to the fragility of themselves, isn't it?
Yep. They do have some form of immunity, but if the cell cultures get infected, then the cells all die straight away. We use antibiotics to try and protect the cells, but keeping infection out is absolutely essential.
Yeah, and so really then I was thinking about ways that could present some of these sort of qualities, and this whole idea that it's a very sort of ephemeral, strange life that they lead in this 3-week-long existence. So this was where what I did was I took the materials that were existing there, which was things like the actual scan, the digital versions of the objects and the speech synthesis, and then I added to them with digitised versions of the lab itself. So that included using, and so that's evolved into the film and an immersive piece, which in the Summerhall exhibition is the augmented reality app, which is just for iPhones, but it's downloadable. And so that was made adding to the existing materials by doing LiDAR scans of the lab environment, which LiDAR scans being a laser technology, which then gives you a point cloud version of the lab. So again, it's a very ephemeral, it's a very, very exact process, as all the microscopy processes are. I mean, with their own artifacts, which is saying, there's all sorts of artifacts that they bring, but they're very exact as well. And it's the same with LiDAR, which it's another form of digitally capturing, but it's not photorealistic, and that was a real sort of aim for me, that it wasn't just actually about a photoreal version of the lab for people to enter. It was to try and sort of enhance that very disembodied quality. That
quite thoughtful, isn't it? Thought-provoking.
Well, that's hopefully what it brings for people. And then on the soundtrack side of things as well, it's added to by an electromagnetic scanned version, which was developed by sound designer Zoe Irvin. So that's again bringing, it's all mediated versions of the environment that are digitally mediated and have these sort of slightly other qualities, again going back to the otherness that I found in the cells themselves. So that's what's then going to be presented as the projection and the app as part of the exhibition. And then it was then to sort of say as well that the other things that are going to be exhibited alongside are some of the things we've got here as well, which some of the things which I was really interested as well around the collection that you've got. Now having moved to the University of Edinburgh from the University of Dundee, and we've looked in your library collection there, haven't we, and found some quite extraordinary things.
Historical dermatology, yeah. Historical dermatology blows my mind. It really is, to me, it's amazing how carefully people observed skin back through the ages, and that's really how medics learned about skin, often by representing it through very detailed drawings. So, yeah, a couple of the drawings that we've brought along are really ancient. Was it
1860 to 1888. This is from the Atlas of Skin Diseases, isn't it? And so this is an eczema patient from that period where, and also you see, I mean, I'd seen these before, but never in the first-hand edition of it.
Yeah.
Chromotypes, I think they're called, the prints. But it was for me, I find these fascinating as again, going back to that idea of the lived experience of things, that actually and as we've talked about a lot in terms of science development, and you working very closely with patients as well, you're very aware of the lived experience of that, but also there's a sort of, it can be quite paradoxical with sometimes the way that research appears to go, that actually, and I think that's what's very special about your position, where you're both a consultant doing work with patients and you're working in a lab environment, so you have that complete connection between the lived experience. So, I mean, I think that's a really special situation.
That's why I do research, because you meet patients that really suffer, so then you have to, well, for me, it's a compulsion to try and understand their skin disease better. But yeah, the historical stuff, I mean, when you look back at it, they are very emotive, I find. You look into her eyes and you can see the suffering. And nowadays, there's still stigma around skin conditions. My patients still suffer misunderstanding and misconceptions because they have skin problems. And sometimes people feel embarrassed or ashamed about their skin, which is something that we really are able to help with. We should be able to put that aside nowadays with the better treatments that we have. But still, my patients suffer, and you can see in her eyes she suffers. Imagine you had a skin condition like that was so difficult to treat. This is actually infected eczema, so with impetigo that we can still get nowadays, but it wouldn't get to that extent. The historical perspective in some ways shows how far we've moved forwards, but in other ways emphasises how good the physicians were there, and how much we still have to learn even now.
And the craft of the documentation in that as well, I think, is that what I'm really, really interested in, as well as how how it actually, as opposed to a lot of dermatology images that you might see now, which actually might be a lot of the time anonymised, whereas that is completely unanonymised in that way. And so actually it's very much putting you into the perspective of that lived experience of that person as well. So I think that's what I found really fascinating, as well as the craft. And then you were just going to say as well about this, there's another piece these are all part of the exhibition as well, aren't they? But it's an early one, that's from 1905, that is looking on a cellular level as well, which is fascinating. I mean, one of the reasons why I took it out, and I think why you were interested as well, was how long, and actually really recognising that this has been for a long time, that people have been looking at this in this very detailed way, which then your work, which we've got a slide of here, which will be in the exhibition as well, is really at the pinnacle.
Yeah, I mean, it's nice to say we're at the pinnacle of research. We use genetic techniques and molecular analysis of skin, but still a lot of it comes down to actually looking at the skin down the microscope and seeing what it looks like. So that's what was being done in 1905, and that's what we still do nowadays. And again, this is a slide from a scientific talk that is totally up to date. It's a talk that I gave recently showing the organotypic culture. This is the artificial skin that we grow, and comparing it to normal human skin, showing that the structures are similar, that the artificial skin does truly form a good model of the normal human skin. So, in some ways we're very clever now. We can use molecular analysis, but in other ways, carefully observing human beings and looking at skin down the microscope is still very fundamental to understanding what causes disease at the deepest level.
And I think this is one of the things that was really key to me and my interest in the organotypic culture as well, and it is part of the soundtrack as well, because it was very much the explanation where you talked, because for me it looks so similar to human skin that would be inside the body, but it's like that sort of smoothed over without fat cells, hair fibres,
Yeah, so
things. So that's, and I found that really fascinating, and you can see it within the slide itself.
yeah, and you can see as well, you know, what we were talking about, the differentiation. The cells at the base are quite plump, and then as they move up, they become more differentiated and become flatter. And this is where the 3 weeks comes from. Do you know that the time course that the skin cell tells the story? It takes a full 3 weeks from extracting the skin from human tissue to recreating the organotypic culture. Takes 3 weeks. And then, you know, in theory, we could keep them alive for longer, but after that time, they start to become less good in terms of structure and function. So at around 3 weeks, we destroy them because they've been incredibly valuable. The images live on, but the living tissue begins to degrade, so it gets destroyed.
It's fascinating. It's really fascinating. And I guess it's that thing as well of really acknowledging then as well, isn't it, the donors as well in that situation. I mean, how valuable they are to you as well, isn't it?
It is literally gold dust. You know, we can't do human research without human beings agreeing to contribute, and patients do that, and people undergoing surgery that don't even have skin problems do that as well, and it is invaluable, and every respect and gratitude to them.
Well, it was a real privilege working in the lab and really having, and I think, you know, managing to be able to build this situation, working really closely together as an artist, working this closely with the scientists. So thank you for that.